Well, thanks. I just wanted to introduce Dr. Sarah Childs, who's our last distinguished scientist today. Come on down. You're the next one on the show. Dr. Childs is a professor in the Department of Biochemistry and Molecular Biology and Alberta Children's Hospital Research Institute at the University of Calgary. So welcome back east. I did my PhD in the University of Alberta, so I guess we'll forgive you for that. There's a bit of a rivalry there. She's a former CRC chair in genetics and angiogenesis and was awarded the Senior Scholar Award from the Alberta Heritage Foundation for Medical Research, and Schultz pardon me, Award for General Excellence from the Department of Biochemistry and Molecular Biology at UC. Dr. Childs did her graduate training at the University of Toronto in the Department of Medical Biophysics, and originally harkens from around here. I think yesterday I heard you're from Dundas. That's right. So Mac was just not on the, no, not going to do that. Too close to home.
Too close.
Yeah. So did her PhD in Toronto and then did a postdoc at the BC Cancer Research Center and then the Cardiovascular Research Center at the Massachusetts General Hospital at Harvard. So welcome, and I have the honor of presenting to you this wonderful plaque.
Thank you.
So welcome, and we're looking forward to your talk.
All right. I think we're ready. Well, here we are at the end of the day, and so hopefully, I will keep to time, so please feel free to shut me down if I'm too long. And hopefully I can, I think I'm complimentary to the rest of the day, so I'm going to talk about development, although I'm going to start off with a picture of a zebrafish. This is an adult zebrafish brain. I find it's beautiful. The vasculature is in red, and smooth muscle cells. So vasculature is in terms of endothelial cells are in red, and smooth muscle cells are in green. And this is so beautiful in terms of its pattern, but it's also incredibly complex. You know, some of the vessels look different than others in the forebrain versus the hindbrain. The smooth muscles in some places, but actually there isn't very much smooth muscle. And zebrafish, as you know, is probably about an inch long. And so my lab actually is really interested in how this all gets set up. Before you eat a bad diet, before you develop all these later diseases, what happens in embryonic development? And at this point, what's really sensitive is actually your genetics, because they're going to set up how your pattern and how your cells are communicating with each other. So I just wanted to contrast the picture I've just shown you of an adult brain, and these are by no means to any kind of scale, and what an embryonic brain vasculature looks like at about 2 days of development in a zebrafish embryo. And you can see, you can almost see the number of vessels in the pattern. And this is what smooth muscle looks like at 4 days in an embryo. So DPF means days post-fertilization, just to tell you. And the number of cells, again, you can make out individual cells. In contrast, this is what an adult looks like when the structure has grown a lot. And so, blood vessels, as we all know, are assembled from different types of cells. The angioblasts are the precursors of endothelial cells, and they luminize into tubes a while before the mesenchymal cells come in. These cells actually then have to, through communication between the two cell types, differentiate into mural cells. Now, I'm going to use mural cells. I know people typically think of smooth muscle cells when they think of the regulation of the vascular system, but there's another cell type of pericytes, which I'll talk about as well. The mural cells, as a general category of the type of cells, then secrete extracellular matrix, make this very strong wall. But there's a period in development where you can actually have very weak vessels because they haven't developed all these structures yet. And so at this point, the vessels are very sensitive to destabilization, so hemorrhage. All right. So how does that actually look in a real embryo? This is a picture of angioblasts before they actually have assembled into tubes. You can just see it's sort of granular. You can see little cells there. And this is a few days later. So this is after they have migrated from this very lateral position on the embryo medially. They have assembled into tubes. They've migrated out. There's very little stabilization of this network, so there's a very intricate network with nothing kind of holding it together other than just the junctions between endothelial cells. So looking at the mural cells, smooth muscle cells and pericytes are actually related. And as I say, you probably are familiar with smooth muscle, typically multilamellar, larger vessels. Actually, in a fish, we don't often get the multilamellar except in the vessels very close to the heart. Pericytes are on smaller vessels, particularly in our brain and in some other organs. And in this case, these single isolated cells are kind of, they're almost spidery. They have lots of processes, but don't necessarily wrap around the vessels, and yet they're very critical for vascular stability. Markers we typically use for these, ACTA2, smooth muscle actin, is usually used for the smooth muscle. And there are several markers for pericytes, but the one that we typically use is this PDGF receptor beta, a molecule. So in my lab, when we started out, I mean, a lot is known in mouse, but nothing was really known in fish, and so we had to develop reagents to start looking at these cells in order to see, in order to later on look at processes that are perturbed. And so both using expression analysis and also transgenic line that we developed in my lab, we looked at when smooth muscle develops. And in fact, it took until 4 days of development. Now, I should say, a fish you may or may not be familiar with, counting from fertilization, between 4 and 5 days is actually free swimming and eating. So it's a very, very rapid developmental process. Smooth muscle is developing right at the end of that embryonic period for the first time. And there isn't much of it, as I say. It's really very restricted, very close to the heart, which is down here. 22 days, we start to see in the vessels around the heart larger amounts of smooth muscle. So again, it's a slow model to develop the smooth muscle, but it does. And here, so sort of looking again, the green here is the smooth muscle transgene. This is an endothelial transgene. And you can see over different stages of development, you get an increase in the number and the coverage of the vessels. But we wanted to know where it comes from and some of the genetics behind it. And here's our problem. If we use transmission electron microscopy, we can actually see that there are mural cells, so there are cells on the wall of the green endothelium here, at 2 days of development. But our marker doesn't turn on. And there are pericytes on brain vessels also at 56 hours, but pericytes and smooth muscle cells, even though they have the same origin, are actually quite different. So what we're lacking is these cells, if you sit there under a microscope and you watch the same embryo all day, they suddenly light up. It's like they just sort of appear. They just suddenly turn on the marker, but they're actually there before that. And so what we wanted to do is to identify a marker that might come on earlier to tell us more about the early process of smooth muscle development. So an intermediate marker. We know that both of these lineages come from the neural crest, and they turn on a gene like SOX10 very early. But that gene goes off in this lineage. It has to go off in order to differentiate into smooth muscle cells or pericytes. Here's our ACTA2 marker. It turns on way late. And although there's pericyte markers, there's just nothing there. And so what I'm going to tell you about, and this is going to foreshadow because I'm going to come back to disease using the same marker at the end of the talk, is about a marker called FOXC1. And FOXC1 we got into through looking at diseases and working backwards, which is kind of ironic, but I'm going to tell it in a linear story in terms of developmental time. FOXC1 is expressed in early mural cells in this boundary, and you find cells that co-express the neural crest marker, and we also find, as I'll show you, that it expresses with a smooth muscle marker. So why do we think that FOXC1 is critical? Well, if you knock it out, there's 2 genes in zebrafish, so this is an allelic series, and this is work from our collaborator, Orden Lehman, who works on FOXC1 a lot. You can see that there's smooth muscle in this area. It's actually the outflow tract of the heart. In all these different genetic combinations, but once you start to lose 3 and 4 alleles of this gene, you absolutely ablate. You still have some muscle in the heart that's lighting up, but you completely lose the vascular smooth muscle. So FOXC1 is essential for early smooth muscle. And okay, so how can I convince you these are smooth muscle precursors? So one of the things we had to do, and it's probably easier to look at this column right here. So Tom Weitzel in my lab, who just graduated, did this. And basically what he did is he did transgenes. He looked at the FOXC1 transgene and an endothelial transgene. He looked at a smooth muscle transgene, an endothelial transgene, and then he did co-expression and basically showed that these little FOXC1 cells sit on the outside of an endothelial tube, like smooth muscle cells, and that they co-express the smooth muscle marker, but not all of them. And so this is part of the argument that the FOXC1 is actually earlier in this lineage than the smooth muscle marker. The other neat thing that he was able to do, and it's a really tricky experiment because the transgenes are really kind of faint, is that he's able to use a time series looking at, these are mesenchymal cells. So these are kind of loose, fluffy cells, really hard to image. But you can watch over time, particularly where the stars are, how this mass of kind of cells with ill-defined shape actually turns into smooth muscle cells that are on the vessels. And then he actually was able to show of all these cells, and he had to unfortunately image with a very large number of them, but these cells actually, like this one here, goes on to express ACTA2. So, and we also looked at the adult brain, and this is actually gets very pretty because this is now a marker that, as you can see here, co-expresses with the smooth muscle actin in the brain. So this is a marker that is on very early in development, before smooth muscle markers come on, and it continues in smooth muscle. Okay, so we have several lines of evidence in terms of its expression, in terms of the knockout, following mesenchymal cells, that this is actually in smooth muscle, which really helps us because we wanted to get at what genes are expressed in the embryonic smooth muscle. But we don't have a marker that's really unique to vascular smooth muscle. And so in order to do this, this FOXY1 gene I'm talking about is actually expressed only in the head predominantly and some endothelial cells, whereas the ACTA2 gene is actually expressed in visceral smooth muscle. It's expressed in a lot of smooth muscle. So we did a transgenic sort using the two markers together. So we wanted double positive cells because these are going to be the vascular smooth muscle cells. So we just used a traditional dissociation of cells, FACS sorting, and bioinformatics and an RNA-seq. And this was really productive. So looking at, probably these are levels of, it's probably better to look here. These volcano plates are very gorgeous. It shows us that endothelial cells don't express the smooth muscle markers and smooth muscle cells don't express the endothelial markers. But more importantly, when we do the double sort, we suddenly got cells, greater enrichment of traditional smooth muscle markers, and we got a whole bunch of new ones. So this is really exciting because we can now begin to tease out, there's a lot of transcription factors in here, we might be able to tease out how you make a smooth muscle cell in the first place beyond the known markers that people have already identified in other species, because for the first time we've been able to look so early. So this is really cool. The other thing I'm going to transition to is function. And so from this transcriptome, we were able to see that there's a large number of genes enriched. Of course, ACTA2 we sorted for and is 8-fold enriched. But these are all channels and contractile machinery. So in this little 4-day fish that's about 1 millimeter long and it's got these very tiny smooth muscle cells that really aren't very big, but you've got a lot of the machinery there already that's able to presumably be functional. And I think this is my next big point. As soon as vessels form, they're functional. You've already got the shear stress, which was already talked about today, of vessels. These are now blood cells running in vessels. This is going the artery, and this is 2 veins coming backwards. And that's already happening at 1 and 2 days of development. Here give a really good introduction, so I won't talk about both the shear stress and circumferential stress that these vessels undergo and the mechanical strain. So this is happening as these Mural cells are coming on the vessels. And so what we want to do is we wanted to look at the ability of the developing vessels as they form to be able to contract and relax in response right now to stimuli. Eventually we'd like to do it in response to flow, but we haven't got there yet. So we're going to look at the brain vessels here for cerebral circulation for various reasons. And the way that we do this is we have our embryo immobilized in a dish. We've got a microscope running, and then we can add, for our first set of studies, we've added either vasodilators or vasoconstrictors and trying to figure out when in development. And we can use transgenic lines for both pericytes and for smooth muscle cells, and we can look at the difference. So what are the results like? So in this little 4-day embryo, that's the end of the embryonic period, there's a dilation that happens in vessels that have these little tiny pericytes on them. It's a very small dilation, but it actually adds to a large potential increase in flow. We haven't actually measured that at this point, but the small dilation is extremely consistent. And you can see here in this overlay, so this is kind of the before and after pictures overlaid, so you can see this very small dilation. Smooth muscle cells don't do anything at this point. So if you look at them, here's our control, and then here's our treated 10 minutes later. These are actually matched embryos. You can see half of them, dilate and half of them contract. So there's a very interesting early confused response as to what these vessels should be doing. And maybe this is even just spontaneous and it has nothing to do with the drug. So they are not able, smooth muscles, not able to function at this 4-day time point. Nabila, who does this work, has these beautiful, these are line scans, so she can measure a vessel repeatedly over and over and over again and then compile it into what's called a kymograph. And you can see that there's examples of constriction and advances of dilation, that this happens at different times. This is very close to when she adds the drug. This is, very steady. It's really interesting to actually look at the timing of it, but at 4 days, nothing happens in smooth muscle. Now, when we go to older stages, so this is 2 days later, 6 days, pericytes stop having any response. They don't dilate anymore. And now the smooth muscle, so that's the pericyte graph up here, and here's the smooth muscle graph, and you can see that there's a constriction that happens, and it's much larger than the types of numbers that we were getting at 4 days. Now we're getting about a 15% change in diameter. And again, here's the kymographs that show that. And we've done that in response to phenylephrine and noradrenaline so far. So 2 drugs. So this is kind of exciting. So what does this mean? So we've got the genetic development and the marker expression, but we've also got the, aside from that, we've got actually development of function pretty quick on. So at 2 days, pericytes begin to associate with vessels, and the mesenchymal cells that will become smooth muscle are starting to associate. When we go to 4 days, our pericytes, which were actually there a lot earlier, apparently can dilate vessels. This is a bit waffly to me. I'm not entirely convinced that they are actually doing anything active here. But we've only got differentiation going on in the smooth muscle lineage. And then by 6 days, pericytes really don't do much at all now, and now we're just talking about the smooth muscle cells, which are beginning to have that function to be able to contract. And we can't find that they relax yet, so we have to go even later in development for that. So, there's lots of questions, and I don't think for the interest of time I'm going to go through these. But, we kind of wonder, when does smooth muscle begin to dilate? What happens to the blood flow when this happens? How much does the ability to contract or relax in early development alter the structure of blood vessels? And so to just kind of go show you the kinds of directions we can take in the zebrafish fairly easily away from what we've done, the collaboration with bioengineer Tina Rinker in Calgary. And we can take our confocal images, and we use 3 different fluorophores here, endothelium, the blood, and in this case we're using nanoparticles for this project. And we can use very high resolution confocal to get the architecture of the blood vessel. And using measured flow rates, we use particle image velocimetry to get the flow rates in the vessels in a living fish. We can now calculate the flow streams and shear stress, and now correlate that. And so we have not done this once the smooth muscle develops. We've only done it in the very early development right now. But we have the very cool potential to see, as soon as those cells get on vessels, well, what happens? What changes? You could do the same fish before and after. And so that's where we're going with that. So it can be, I think, in the future, really interesting. And so, yes, what happens when the smooth muscle cells adhere? So the other thing I just want to kind of mention with this, and maybe you've probably already thought of that, is that we're really talking about microvasculature and everything I've talked about because the fish is so small. And what's neat about the microvasculature is it actually doesn't vary much across species because it's set by the dimensions of an erythrocyte, and erythrocytes don't really change from organism to organism. So the smallest vessels actually have the same dimensions. So we're not going to talk about large conduit arteries because, quite frankly, that would be about the size of the fish. So that's not. But we have an excellent model for learning about microvasculature and microvascular regulation, the sort of resistance arteries. And so one thing that Tom did when he was looking at development was look at the size of vessels that either showed just the transgene for endothelium or the transgene for endothelium plus a pericyte, or a transgene plus endothelium plus a smooth muscle covered either the FOXC1 or the ACTA2 markers. And showed that, first of all, over development, the size of the base vessel actually gets smaller, which would be consistent with development of tone and some kind of muscular wall. But between even an embryo and an adult, actually the largest vessels don't vary that much, at least in the brain. The larger vessels close to the heart will get bigger. So this is very interesting. And so I want to focus again on brain vessels and tell you a little bit, bringing some of our early development and markers to actually talk a little bit about disease. So I went through a few weeks ago and curated all, at least all that I could find, human genes involved in vascular malformation, aneurysms, and strokes, and classified them kind of, these are endothelial mostly, and then these genes affect like, extracellular matrix and cytoskeleton. And one cluster here is associated with cerebral small vessel disease, including this FOX transcription factor that we actually used as a marker for smooth muscle early on. And we actually work with this FOXF2 gene as well, and the collagen 4A1 as well. So cerebral small vessel disease, I'll introduce a little bit in this slide and the next slide. Let me just put up the model. So cerebral small vessel disease is actually poorly understood. It's actually a factor in a lot of ischemic stroke, but it comes from a different kind of mechanism where the small vessels in the brain progressively either occlude, so there's little things like microbleeds or lacunar infarcts, and it leads in the end to white matter dysfunction. And so you can see on these MRIs that there are sort of white patches, and actually these are 6-year-old children. So this is a human genetic disease caused in this case by loss, duplication, deletion, and mutations in the FOXY1 locus. Again, this is out of Orden Layman's lab, and we've been collaborating for a number of years on this. So, white matter disease, cerebral small vessel disease associated, and we have fish models. So when you have a fish model that lacks this FOXY1 gene, which is, we know, very important for smooth muscle development, you have a loss of vascular integrity. We also work on the FOXF2 gene, very similar to FOXY1. They're actually located in the chromosome right next to each other. It also is expressed around vessels and expressed in brain pericytes as well in adult fish. We don't know in humans, but we do know in mice it's actually expressed in the same locations. And we were able to show through FOXF2 mutations that we get a reduction in smooth muscle in the brain. So this is using a transgenic line to look at smooth muscle. And then we classed it into different groups, and we see that in mutated fish, there's actually, most of the fish fall into this category, so they have a lack of that. Why is that important? Well, through a number of collaborations, this is the Metastroke collaboration. The cohorts in healthy aging, it's out of the Framingham Heart Study, had actually looked at 80,000 participants in their first pass, and then they had a sort of a validation cohort. So there's about 160 or 170,000 people with stroke, and they're not looking, it's just all strokes. They're not necessarily breaking into categories at first. And what they find is that there's polymorphisms right near the FOXF2 gene, that is the one that we're working on, that correlates with all stroke. And they found, actually in this study, they only found 2. They've now repeated it with a couple 100,000, and this gene still comes up. So our developmental work that kind of looks at markers and how does this lineage develop actually is coming full circle with, defects in these very early genes actually probably segregate with disease, but we have no reason why, right? Correlation is not causation, right? These are SNPs in the genome. They're not protein SNPs. They're actually SNPs in the intergenic region. And so part of my lab is actually working on taking that human genetic region and trying to figure out if it's an enhancer for this gene. But how might it work? So I've said these genes are important for the development of the lineage, but can we look at any kind of function? And so what we've done is sort of, again, coming full circle in our functional work, we can now test in our animal models contraction, relaxation. Is there any defects in this? Is there any defects in collagen deposition and other factors? And so we've been able to show wild type, I've shown this data before, wild type pericytes can relax at 4 days in response to sodium nitroprusside, nitric oxide inducing agent, and this is what the general graphs looks like, and there it is. And our preliminary experiments with our mutant model say that our mutants are incapable of the same function at the same stage. So, could, we really, as I say, we really don't know how cerebral small vessel disease works. But what we do know is that we have a couple of genes that are associated with cerebral small vessel disease that are expressed in Mural cells. We think they probably influence expression of collagens, but there are other things. There's something wrong with them in terms of how they can probably regulate blood flow. And so the kind of the exciting, being able to work in a developmental model may help us tease out some of our models are probably far more. We're doing knockouts, and in humans this would be a small change in expression. But it allows us a little bit of a window into what do these genes do and how could this cause disease? And if it causes disease, is there any way that we could eventually fix it because we kind of know a little bit more about the mechanism. So to kind of take it to sort of summary, I mean, what my lab does really is take genes. We do look at the developmental origins, so we do a lot of microscopy and tracing and imaging and development of markers, and we've got other markers as well. But we'd like to take that to a functional level, and we're in our infancy in being able to do this, but I hope I'm recruiting some computational people, and hopefully we can take this to a higher level to understand smooth muscle function. The blood flow is a high area of interest, and so we're going to start looking at the entire, if we can do this. It takes 3 weeks to do these computational models, 3 weeks of computer time currently, so we have to either find a bigger computer or a smaller problem. But look at how the whole cerebral circulation is in a mutant versus a wild type or under different drugs, or we can do lots of manipulations because we work in a genetic model. And then disease modeling. So we happen to have in our lab a couple of models for various diseases that are poorly understood. And so trying to put all these different parameters together. So I hope what I've shown you today is that we can model genetic mutations leading to stroke and vascular malformation. Now the ones I'm working on are very non-canonical and new, so we're sort of basically starting from scratch, and I don't think people would think they're accepted stroke models, so I think we have to go through and show that in aged animals we will get stroke, and that we can really model a microvasculature. That our FOXC1 is an early smooth muscle marker, and it's going to highlight, for us, what it highlights is the very early process of attaching of Mural cells to the first endothelial tubes, and that we have a model in this FOXF2, sorry, in the two different genes, too many foxes, I know, but it has potential defects in these small vessels, particularly in the ability to dilate, and we haven't tested contraction yet because we're kind of chasing the wild type animals and trying to figure out what they do, and then we have to go back and apply it to the mutants, so we haven't got there yet. But we're hoping that these and the other models we have will help our understanding of genetics, and really you can see this kind of pipeline could apply to a lot of things, and the function of the cerebral small vessels. So in conclusion, I'd love to thank my lab, particularly Tom Whitesell, and Nabila Barami does all the functional. Danielle Blackwell did some of the functional as well. And our collaborators, Orton Lehman, very strong collaborator, University of Alberta. Peter Carlson works on FOXF2 in mice. Stephanie and Suda were our collaborators in human genetics, and Nathan Lawson did a lot of bioinformatics, and Tina Rinkers are bioengineering. And I'm looking for someone who is expert in computational fluid dynamics to work on some of our bigger problems we have in this field. So thank you very much. Happy for questions, yeah?
That was a beautiful presentation. I'm sort of wondering, in terms of pressure gradient in the fish, so what kind of gradients do you expect in these microvessels?
So actually, some folks have measured them, and obviously compared to a mouse or human, the blood pressure is much lower. I mean, they're in an aquatic environment. They, water all around. They're actually swimming horizontally, so they don't have the same sort of thing as us. So they're going to be very small at the moment. The other thing is it's dynamically changing, and so one of the things we've noticed is that when you get those hemorrhages, you get that vascular instability. The time it typically develops is between the first and second day of development, and what I think is there's a big step in pressure that happens. So, at first you're doing just a simple circulatory loop from the heart to the tail and heart to the head. And then on the second day, you're suddenly putting all these microvessels in. And that's when it's like, poof, so I would love to measure it, but I personally haven't measured it. People have, yeah.
So in terms of the vessels, you see a lot of endothelial vessels, you know, like it's a sort of capillary type
Yes.
formation, right?
Yes.
And so the smooth muscle, you get one sort of single cell smooth muscle, or you get like thickness to the
So at the stages I'm talking about, it's only a single.
single cell.
It's a single. And it's, as I say, it's like 22 days to see that multimolamellar smooth muscle around the outflow tract of the heart. So we're talking, you know, it takes a long time. So as I say, there are going to be differences between mammalian models with much stronger pressures and more rapid development. But I think, you know, at some level, understanding that process, right, just the mechanics of how that happens will be applicable across organisms. Yeah.
Well, let's talk to you. In cerebral small vessel disease, do you notice that the microbleeds are more prevalent in certain brain regions?
So I don't know in a human. I don't know at all. In our fish, it's usually, it's not hindbrain, it's midbrain, forebrain. And what we know about Mural cells, and pericytes in particular, is that midbrain, fore and midbrain are actually derived from neural crest, whereas hindbrain are derived from mesoderm. So it's probably a problem with the neural crest derived pericytes. But I can't translate that to human, sorry.
Yeah, so you don't think it's the flow pattern changes?
You know, and again, I don't know here yet what the flow is. That's really what we've got, where we've got to go. Absolutely, and which vessels are the ones that I do know. And I should have showed these, but you know, the videos take time. We do have a repeated imaging, time-lapse imaging of a hemorrhage developing in fish, and we've done that many, many times. And it's the small angiogenic capillaries that are bursting. So it's not the large vessels, it's the small ones, but I think it's when the flow, it's really neat. When you watch it in real time, you can see erythrocytes coming and this poor little capillary isn't open. The erythrocyte will push, like pulsatile, push, 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 and eventually it'll make its way through. But I think if the tip of that isn't ready, it'll burst, and so that's what's going on for those, yeah.
So as soon as you, that remarkably, you mentioned that as soon as there's the vasculature, there's blood flow already.
Yep.
So how does that work? I mean, what's the rate limiting? Is it all ready to go, or as soon as, or reverse? What's the rate limiting factor?
So the blood is forming, so because the blood is a component of this, right? The blood is forming really at the same time. So there's a region in the aorta where the blood is forming. The tubes for the largest tubes, so the aorta and the posterior cardinal vein and some of the carotids and things like that are forming by vasculogenesis. So they're forming very early. They're not the sprouts. So it's a different time of angiogenesis. Why? I think that the pressure is really important for the formation. If you don't have blood flow, people have shown that you still get sprouting, but they're very, shall I say, weak-looking sprouts. They're very spindly. They look very different than if you have a full pressure. And so I think that mechanosensing, people have shown that notch signaling is turned up by the flow, so it's another mechanosensor. So I think, and BMPs as well. So there's BMP that comes from the heart, secreted by the heart. It needs to run through the circulation. So I think you need it to get the further development. You need that flow. Yeah, from a couple perspectives, yeah.
So Sarah, you mentioned in that cerebral small vessel disease that you sort of hypothesized that FOXF2 might be regulating tone.
Well, I don't know. That's a hypothesis.
Do you have any evidence that it might be adhesion? So adhesion of the neural cell to the other?
Well, so we have the sort of the, you know, when you look at the population of animals, they have poor smooth muscle appearing on their vessels, and so that could be an adhesion defect, right, because they're not getting there and they're not sticking on, and if they don't stick on, they don't express the transgene. We're looking at ACTA2, so they're not maturing. So it could be there. And we have looked at collagen 4, and definitely collagen 4 is much down. So, and whether that's reflective of the fact that smooth muscle cells haven't come in, and if they don't come in, they're not spitting out collagen because they're a huge producer of the collagen 4. So yeah, I have some evidence. And so what I'm kind of going off is that Notch3 is another cerebral small vessel disease gene, and some people seem to think that actually the vessels are stiffer, and that's part of the problem. So they're not able, again, to properly regulate tone. So I'm kind of modeling this because we think this might regulate Notch as well. So there's lots of things going on, lots of stuff to find out, but I don't know yet. Thank you.